you are the first, yes.
Because I sneaked in early and sorted it. If that's the case, hang on. If that's the case, because we've got time. Oh, we're not connected.
Okay, so to conclude the Amble session of this session, the next speaker is going to be Andrew Nightingale, who is going to talk about proteins and protein services in order to integrate and interpret UniProtKB. Thank you.
Hang on, excuse me once I get rid of one thing that I can see over my slides. Once again, thank you very much for the organisers for giving me the opportunity to speak here today. Yes, it's very strange that we had been blocked in as an EBI tech track specific stream. So I'm Andrew Nightingale. I work in the UniProt group as a data scientist, and today I'm going to talk a little bit about UniProt and some of the updates at UniProt, and then much more about the data services that we're starting to provide to help you use our data in your own websites and to interpret your own information. Excuse me, now that I've turned on the Wi-Fi, it keeps on coming up with a thing to connect to it, so I'll have to stop and be distracted every single time. So yes, I will first of all give you a quick update on changes in the UniProt website. These will be as a lead-in background to the rest of the talk, which will be about our services and technologies and how you can access UniProt Knowledgebase function annotation, specifically through Vista and the Proteins API. Then I'll talk about some new libraries that we have, which you'll be able to use, and we already use, to have the data and use some visual components, and then show you a couple of applications of this technology in services which we are developing at this point in time and will be available soon. So for those of you who don't know much about UniProt, UniProt is a large database, which principally the information that we provide comes from the UniProt Knowledgebase, which is comprised of SwissProt, which is, has about 560,000 manually curated entries by expert curators. And this is a non-redundant database and has a large number of functional annotations and cross-references. And then we also have a much larger set of sequences, 160 million sequence entries in the Tremble data set. And this is redundant. It is the set of computationally derived sequences from Ensemble and other genomics resources. And this has some automatic annotation in it. And the automatic annotation is driven from the knowledge base from the SwissProt dataset. And when you look at the graphs on the right-hand side, as you can see, SwissProt grew very quickly at one point when the reference genomes were being completed, and is now plateaued a little bit. That's mostly because what we're doing is continuously updating the protein entries and adding in new functional annotations and improving the functional annotations. So that's not growing very, very fast. What is still growing very fast is the Tremble database, where in 2012 we introduced a proteome redundancy procedure, which at that big crash down reduced the number of Tremble entries we had significantly. And we started growing linearly for a little bit, but now we've started to grow exponentially again. This is in the context that we still are actually running the proteome redundancy. And this is mostly because of the big sort of like Terra Ocean projects, which are bringing in large number of new bacterial proteomes and genomes. And so we're still picking up all these sequences. Few announcements. Firstly, UniRef clusters, if you use UniRef clusters, the way they're generating is going to be changed in the 08 release of UniProt. We're moving to using the MM62 clustering algorithm. This is because of a number of reasons. One, it has a much more improved sequence alignment algorithm over the current method we use, much more advanced clustering framework, so the clusters will be a lot more robust and better. And due to the exponential growth in the Tremble database, it gives us the greater ability to optimize, to be fast in the clustering because of its optimization and speeding of the clustering, and the fact that it is parallel. so we can compute everything in parallel. And this is in the context that we expect to be growing even more exponentially with the number of sequences we get into Tremble. For you, you're not going to see any differences. It's much more what's happening in the back end, and it's just to introduce you to the concept of actually how these clusters are generated. If you want to become a contributor to UniProt, we have a prototype where you can actually add in specific publications to an entry. And this, up here it describes to you, all you have to do is come in through, retrieve the protein entry, sign in with your ORCID ID, and then fill in a submission form to retrieve the publication, add the annotation, and then it will be submitted. Why would you want to do this? Well, because you might be an even greater expert than our expert curators on this protein entry. And it will help us scale up some of the annotation and curation effort of the proteins and potentially add in new protein entries from the Tremble. And it will provide a much more comprehensive set of articles related to that protein. What do you get? Well, you get credited for actually annotating and putting in those papers. Your contribution is citable, so you will be able to cite that, and it will, you can play a much more interactive role in improving the UniProt database. So if you're interested in actually adding in some articles, please go to the link that's below and try it out. Next, we have standardized the way we represent enzyme annotation. In the past, what we had was a free text definition of the enzyme reaction. This is now being completely redesigned. We are using the standard RIA reaction database sets. The compound sets come from Chembl, and this then allows us to have a much more comprehensive definition of the enzymes and the enzyme reactions. And why have we done this? Well, because 50% of the entries in SwissProt are actually enzymes. So this is an important set of protein entries, and providing this information in the correct way is essential for you as users to be able to use this information. And along those lines, now that we have updated the way this works, you are able to do much more complex queries on this information. You can search for enzymes that react with D-glucose, for example, or which enzymes are in a particular pathway, et cetera, et cetera. This also leads on to the point that finally we have managed to link all the omics information. So, excuse me again. So you can now go from a genotype to a metabolite, and you have the full link from genotype to the metabolite phenotype with this information, with UniProt being in the middle at the protein section. So hopefully that's given you just a quick overview of some of the changes that have happened in UniProt, and given you a bit more of a foundation of what is happening in UniProt. What I'd like to do now is just move on to the services and technologies. If you've ever been to the UniProt entry page, which is the main entrance to UniProt, you will find a sort of page like this, but this is a bit more of a sort of like rearranged to show you information. And so apart from UniProtKB, we obviously have UniRef, UniPark proteomes, and below that we have the programmatic access points. And if you look under UniProt data, if this will work, thank you, you will see that we have a specific link to programmatic access. And when you go to that page, you see that we actually have 4 different ways of programmatic access. The main principal one really is the top one, which is the UniProt website REST API. Whenever you do advanced queries in UniProt, or you do a basic search, you find that you're actually using that website REST API. And if you have no experience in programming, that's a good way of starting to build up your experience. Alternatively, there's the UniProt Sparkle API, and for more advanced programmers, there's the Java API. Today, I'm going to be talking about the Proteins REST API, which is another API that we provide. This is available separate on a Swagger website for you to try out. There's one thing I've marked up in red here straight away. Up to very recently, we didn't restrict your access. We've had to restrict your access to 200 requests per second per user. If you want to do an awful lot of data, there are ways of getting bulk data down. If you go to the example section, you will find ways of doing that, okay? And if you have any questions, there's also ways to contact us and we can explain how you do it. You do not need any programmatic experience to use this. This is a nice interactive website, exactly like the EBI website, search sites, where you can look at what you can put in different search fields to look up certain information, and you can just click on a tryout and it will return the information for you, okay? And this actually supplies slightly more data than you get traditionally through the website REST API or the Java API. You get additional information like genomic coordinates and a fuller set of variants, not just the reviewed set of variants. You get a lot more variants from the likes of TCGA, 1000 genomes, exact, et cetera, et cetera, et cetera. And simply put, if you go down, there's all the different types of access points on the right-hand side if you use this Swagger page. Once you get more experience, you won't necessarily come into the Swagger page. You'll be doing everything programmatically. And I say that because once you get used to this, you may want to be choosing a very specific sort of file format. And we provide the standards, XML and JSON. Also, because we're a sequence database, we have FASTA and GFF. And we have the new PEF format, which is the proteomics extended FASTA format. We are only supporting at this point in time for the proteomics community the variance annotations in that format. As this format gets more established in the proteomics community, we will add in additional functional annotations. Okay, from that point, all of these endpoints here actually feed into a lot of the stuff that you actually now see as interactive information on the website, specifically Prot Vista, which is the feature viewer that you see when you go to a protein entry on the UniProt website. This is a fully interactive visual, tract-based visual interactive site that allows you to explore all the functional annotations and look at various things. We've added recently also the structure component, so you also have the structures. If there is a structure, you can link that through, and I have an example of that a bit later. What we're trying to do now is actually take all these combined tracks, which are in the Prot Vista, and split those out into separate components that you can use as embedded separate tracks at specific locations within the UniProt website or within your own website. And to do that, we've created a set of new libraries, a specific library, which is called the Nightingale library. Note the asterisks after Nightingale. There's an explanation why it's called Nightingale below. If you want to know the relationship to the family name, I'll do that offline later. All right, this was not deliberately named after me, okay? So this is a library of reusable data visualization web components. Web components are a new internet standard for allowing you to have actual JavaScript elements and interactive visual components within your own website. And they've been really starting to be established in the last 18 months. The only browser that doesn't really support this as well as the other ones is now Internet Explorer. If you go, as I will explain, you can go to where our library is, and there are ways to explain to you how you actually get everything to work in Internet Explorer. What we're trying to do here is create a set of flexible components that allows us, firstly, to represent all our data in an easy way, but also allow you to use our data and extend it to your own data. Okay? And these components can be used anywhere within HTML. You can use them using native JavaScript or something like a framework like React. And this is the only bit of code that I have up, okay? It simply, within 10 lines, you can get everything working and get a track up and running. So this is all available for free on the Nightingale library in GitHub. There's a simple get started which will talk you through how to upload the library and get it started. This is community-led, so Interpro are also contributing tracks to this, so there is an Interpro track. Here is the manager example. I'm going to put this one up. It's a bit more complex than some of the other ones, but it will really show you how to lay out everything. And it looks a lot like Prot Vista, so you're starting to understand how things work. And we are creating a new alpha release version of the Prot Vista, which will be based on the web components. There is already Prot Vista for PDB, which has specific protein structure tracks, which is an extension on. And again, that is available under their GitHub if you want to get it. So to finish off, I'm just going to show you some quick examples of how we're using web components currently in the website and how we might use them in the future. So if you go to the structures section of the web of a UniProt entry, instead of having a list of all the PDB entries that are available and mapped to that entry, we now have this interactive web component, which uses both the Proteins API and the PDBE API to load up data, so you can interactively play with the structures, look up different structures that might be of interest to you. I'm getting distracted by. And so you can see that this is using light mode to load up. This is all real time now. I have not quickened this up. This is how responsive it is about actually doing everything. And finally, as an example, I'll quickly talk about the disease web pages. These are a concept that we're working on, and so I'm not really going to explain too much about actually the structure of the disease pages. These are just a slightly different view of the UniProt data and a compressed version. But with using these, what we've done is we've been able to link additional information so you can navigate through. And here is the example of if you are interested in the variants. And so this is a new variant track here. This one's specifically for the UniProt reviewed variants, and it will just simply list at this point in time the diseases that are associated with those variants. And also, because we're components, you can do lots of things and you can embed lots of stuff. We also have the pathways. So this is the Reactome pathway visualizer, if you've ever gone to Reactome. And so this is embedded in a web component, and you can see that it's still fully navigatable, and you can quickly navigate through that to understand the pathways relationship. And to finish, Peppep is a new service which we're providing. It's in beta at this point in time. This is a collaboration between UniProt, Ensemble, PDB, and the Janet Thornton Research Group. This is using all of our REST APIs to bring in all the variant data that is available. This is the first time that we're trying to actually integrate all this data so you get a full genomic view and a protein view. You get the individual variant information, but you also get the overview at the full protein class. The current site looks a bit like this. If you've done a search, you get a results page like this. When a variant is in the gene, you get obviously impact on the protein. We show them based on the canonical, but if you want to see the individual isoforms, you can see the isoforms. I'm not going to show that here today. We give you a CAD score, which gives you a very quick overview of the impact of that variant, so red and green. If you want to understand how those color schemes are working, you have to go to Ensemble, because this is Ensemble's coloring scheme. We provide various impacts, genomic and transcript, protein function and structure function with co-located variants and if they're disease associated, a clinical impact, and you can download all the data. And if you actually go into this and you do it, you'll get a summary page, but there's also a details page which has a lot more information about the variants and is also interacted, which is currently provided by the VAR site. And just to finish, acknowledgements. I work within the Maria Martin team, which you can see in the picture there, but obviously UniProt is a large consortium of 3 groups, and none of this is possible without all 3 groups, and that's the entire set, plus our funding. And the links that were, as I flashed through, if you want to take those down, by all means do so. now as I take some questions. And thank you very much for your attention.
I was wondering, you were saying that contributions to UniProt were citable. How do you enable that?
Right, so it's a prototype. Sorry, I'll just sit forward. The group at PIR who are organising this are much better at understanding this. I was just doing the announcement for this. And what will happen is, I assume what will happen is, as you do it, that interface will then pick up that reference you've done, what you're citing, and then that will be automatically added into the actual protein entry. Okay?
Now we'll move to the next part of the day, which we have a 1 hour talk from IBM about IT infrastructure for AI and analytics in medicine and research. And our speaker is Ulrich.
So you need to have the presentation probably on your laptop.
No, I don't,
Okay,
because